Good. Hi everyone, welcome to the exosome presentation. Today we will highlight key variables that impact exosome isolation success. By the end, you will have a clear view of the exosome landscape and how to refine your own workflow. Quick question to the audience, where is everyone joining from today? Just type your country in the chat. Before we start, let me introduce you to Eddie the exosome at Norgen. Today, Eddie is hiding throughout the webinar. You can count how many times Eddie has shown up and make your best guess in the survey afterwards. Eddie is hiding throughout the webinar, and you will get a chance to win a gift if you get it right. So this is Eddie's first appearance. Let's see if Eddie will come back soon. We'll begin with a quick introduction to our speakers. Today's presentation will be led by Maggie Xue, Project Manager at Norgen Biotek. Maggie brings a strong academic background in biological sciences from the University of Guelph and supports our scientific teams here at Norgen. The Q&A session will be held by Dr. Chauhan, Lab Manager and Research Scientist. Dr. Chauhan is a molecular biologist with a Ph.D. in cancer biology and over 5 years of experience specializing in exosome research for cancer biology and liquid biopsy. Since 2020, he has served as a research scientist at Norgen Biotek, where he's led and supported projects in exosome isolation and nucleic acid profiling. Uh-oh, looks like Eddie's back on screen. Don't get too comfortable, it only gets trickier from here. Now I am passing the mic to Maggie.
Hi everyone, I'm Maggie, the project manager here at Norgen. I'm really glad to speak to people all across the world about exosome. And here I'm checking the chat right now. I see this answer keep coming in. I see people come from Australia, from UK, Canada here, and Dubai. Well, welcome everyone. Come to my presentation today. So I believe everyone who's interested in this topic, you probably already have some background knowledge in exosome. But for those who don't, I will start with a quick 1-minute refresher before we jump into the main topic today. So about extracellular vesicles, such as apoptotic body, microvesicle, and exosome, they are the tiny membrane-bound particle released by the cell. And among those, exosomes are on the smaller end as indicated in the right picture here. Exosomes carry a variety of cargo specific to the cell of origin, include specific DNA, RNA, protein, lipid, and metabolites. Due to exosomes' unique cargo-carrying property, exosomes, they are heavily involved in cell-to-cell communication, regulating the immune system, tissue repair and regeneration, and modulating inflammation. Therefore, exosomes are great biomarker for detecting diseases and cancer, and they can also be used as a vehicle to carry drug in regenerative medicine. And in addition to their wide clinical potential, they also become popular in cosmetics. Okay, so before we move in today's topic, I want to ask, has anyone ever struggled with figuring out what's the right exosome isolation method? You can simply leave a thumbs up in the chat if that's you. Okay, so that was a really brief overview of exosome. And before we're isolating them, well, of course, we need to pick the most suitable isolation method. But as Exosome Isolating Pro, we think beyond that. In today's presentation, we will take you through the 4 factors that a pro consider before isolating exosome. Include the type of the starting sample you're dealing with, the isolation method, of course, the end goal for isolating exosome, and the 4th point, well, that will be reviewed near the end of today's presentation. And you can also check your knowledge on how much you know. If you know everything from today's presentation, well, you are an Exosome Isolation Pro certified by Norgen. So I'm checking the chat over here. I think there's a thumbs up keep coming in. And honestly, it is totally relatable. Because choosing the right method, it can be tricky. And that's exactly why Norgen, we are here to help you today. So without further ado, let's dive into the first factor, the sample type. So exosomes are present in conditioned media and virtually all bodily fluid. They could come from some common samples like cell line, blood, saliva, or urine, or from the novel ones like plant. And each sample type, they come with unique challenges. And here, I have a quick question to the audience again. I'm wondering, for those who are involved in the exosome field, what are the sample type do you usually work with? Feel free to let me know in the chat box, and please surprise me if that's something that's not mentioned in the list here. I'm waiting to see. Well, for today's presentation, we will focus on some common ones to give you an idea. And the first one we will start with is the cell culture media. Well, cell culture media is widely used in exosome study, and it allows the researcher to study exosome in a controlled in vitro environment. So MySIF 2023, it is the collection of the current broad consensus in EV research published by ISIF. And according to MySIF, here's the list of a parameter to consider when when cell culture media is your starting point. So you should consider detail about the producing cell you work with, the media component, the cultured condition, and etc. But there is one thing I want to talk in greater detail today. Is that MySIF point out that the cell culture condition actually directly or indirectly affect EV yield composition and function. Why? Because the component of the cultured condition, such as fetal bovine serum, they not only contain EV, but they also are rich in them, which can introduce bias into EV-related research. So here are the 2 options. We can either deplete EV from the supplement, or we can use commercial EV-free product, and that is where Norgen comes in. So at Norgen, we provide a solution to this problem, and this is the first Norgen product I want to show you today. So Norgen, we have the FBS exosome depletion kit that can deplete exosome in FBS in just 40 minutes with a flexible input up to 280 milliliter. And you can see from this comparison chart that with the exosome depleted, the FBS and the standard FBS, it shows similar growth and identical cell morphology. That means the function is ensured without the FBS exosome getting away. Okay. So now I'm checking the chat, and I see a good amount of people doing blood, and few of them dealing with cell culture M.Sc. And there's one urine folk. Well, welcome, because that really helps us to go through more tailored tip. Because the next one I want to mention is blood. So for blood, it is widely used in vivo exosome study. And since blood, it reflects exosome from the circulatory system, it carries a more systemic relevance. And therefore, blood can be used for biomarker discovery, for diagnosis, prognosis, and therapeutic monitoring. And among all the bodily fluid, blood is the most studied one. And from myself again, it is suggests that when we are dealing with exosome isolated from blood, we need to consider the variability in donor composition and pre-analytical processing variability like blood collection, preparation, handling, storage. So blood, it is largely stored and transported by the freezing after collection. And myself advise to remove the cell before any cell disrupting process like freezing and thawing. Well, also exosomes, they are located outside of the cell, right? It is in a cell-free environment, and it is supposedly can be separated from cell just by low-speed ultracentrifugation. However, the red blood cell, they may lie during blood collection and processing, and they will release the internal content such as hemoglobin, and those are the things that will turn the plasma or serum to a reddish color instead of yellow. And well, luckily, that's not gonna happen if you preserve exosome with Norgen's blood collection and preservation tube, which it allows 7 days room temperature preservation that avoid freeze and thawing, and therefore we will not cause hemolysis. And you can see clearly from this picture over here that after room temperature shipping for 7 days, there's no hemolysis happen, whereas it does happen to our competitor and the EDTA tube as indicated here by the loss of the serum, which is the yellow color. Well, I'm not going to detail today for all the sample type due to the time constraint, but the cell culture media and blood, they are the two biggest sample type used in the in vitro and in vivo study, well as also as what I can see in the chat here. For the rest of the sample type, you can check myself. You can check myself for more info on the specific ones you are work with. And by the way, Norgen, we actually have a validated and complete exosome study workflow solution for cell culture media, blood, saliva, and urine. And those isolation kits, they're designed to specifically cater to the nature of each sample type. So you don't need to worry about the pre-analytical consideration. But, well, how does Norgen do it? Well, that leads us to the next topic, the isolation method. And let's kick off the second session as well with a question. So here's the question to the audience. I'm wondering, what are the isolation methods you currently use? And of those, which one do you think is the best? Feel free to type it in the chat box and let me know. Well, for today's presentation, we're going to focus on the 5 parameters, the throughput, purity, speed, cost effectiveness, and yield, to give you at least a base level understanding of the method listed here. We will go through just 5 most commonly used methods, and of course, I will save the best one to the last, Norgen's silicon carbide method. So why don't we start with ultracentrifugation, which is the most widely used method and so-called gold standard. It is density-based and rely on adjusting centrifugation speed to separate exosome from other particle. Well, as a simple and a cost-effective method, the trade-off is that it can take a long time and can co-isolate contaminants. Also, due to the high shear force, it can potentially disrupt exosome structure. And there are some variants of ultracentrifugation that come with improved feature, like differential ultracentrifugation and a density gradient. But for today's presentation, I'm not going into too much detail. Okay, so I'm checking the chat right now, and I do see the question coming in. Again, everyone, feel free to type your question in the chat box. We will answer them all at the end of today's presentation session. And I see there's people mention ultracentrifugation, use commercial isolation kit and SEC. Well, that's great to see such a good mix. And of course, really there's no wrong answer because all of the methods, they all have strengths depending on your goal. So the next one I want to mention is the ultrafiltration. It is a science-based method that uses pressure to push samples through a membrane with a specific pore dimension and molecular weight cutoff. It is cost-effective and quick, but the pressure can damage EV and the membrane adhesion may cause sample loss and aggregation. And also, there are many improved versions of this method, such as sequential ultrafiltration and tangential flow filtration. And another size-based method is size exclusion chromatography, which the sample passes through the part of the stationary gel. This method is quick and reproducible, with the major benefit being that it preserves exosome integrity and bioactivity. Well, however, because a similar size component, they are not effectively separated, and special equipment, they can be expensive. Well, we're halfway there. Thank you for sticking with me. And here we come to the polymer precipitation, as someone mentioned in the chat today. Well, this use a hydrophilic polymer to bind water and chemically precipitate the exosome. Well, it is easy, it is inexpensive, and it does not require specialized equipment. And also, it comes with high yield. But the trade-off is that sometimes it has a low purity due to co-precipitating the contaminants. And this added polymer, it could sometimes interfere with the downstream analysis. And the last one, before we start introducing the Norgen solution, is the immunoaffinity. I believe someone also mentioned in the chat today. So the immunoaffinity method uses antibody or ligand to target a specific exosome surface protein. And this method is by far the most specific and yield the purest exosome. But the trade-off comes with that is it has a low yield since it only captures exosome expressed at the target marker. And also it comes with a high cost, so it only really suitable for small sample volume. And finally, not just silica carbide method. Well, it is a pH-based adsorption method used our proprietary resin. And the process is just simple. So we adjust the pH to the isoelectric point of exosome surface protein to enable efficient binding to silica carbide. And then the impurity are washed away and exosome they are released by adjusting the pH again. And all this process is only within 30 minutes. Well, I was going to make a spider chart, but I know you probably wouldn't believe me if I rate Norgen a 10 out of 10 across the board. So instead, I will show you the data and I will let you to be the judge. Okay, let's get started. So in terms of sauropod, Norgen will come with a slurry format, which it is in a liquid form, and that allow easy integration to high sauropod workflow. And in terms of purity, here's a publication that highlighted the principle and problems of exosome isolation method. And they mentioned that Norgen's method is fundamentally novel and result in considerably low level of contaminating material, especially compared to EV isolated by ultracentrifugation or precipitation. Even when we compare to other commercial kits, you can see that Norgen used the highest level of usable marker protein and RNA, along with greater reproducibility. Okay, so since purity and yield, they are actually conflicting concept. As purity, it means cover selectively, and yield means cover more broadly. Our goal here at Norgen to strike the balance is that we are looking for the highest yield from the purest exosome. And this is validated in another publication here. So in this publication, they compare exosome isolation commercial kit for human saliva. And they also use immunocapture to confirm the true RNA content. So this is the picture before immunocapture. And you can see among all the methods, Norgen showed the lowest concentration. How come? Well, after validation, you can see that Norgen actually has the highest verified exosome concentration, and in conclusion, Norgen achieved the highest percentage of real EV recovered. And lastly, I want to mention cost and speed together. Norgen isolated exosome in under 30 minutes at an affordable price. And we know time is money and money is money. So this saves you on both. And if you're interested, feel free to check out our project with the QR code list here. And I will move on in 5, 4, 3, 2, 1. Okay. So you might have your favorite method in mind, and I see a lot of them listed in the chat here. But the reality is there's not a single best method out there. Because really, there's not a single parameter, whether it's purity, yield, or speed, that should be universally prioritized. And instead, the choice of your isolation method, it should reflect the demand of your application and the research objective. So here's a rough breakdown of what people are using exosome for. It's either for analytical purpose or for therapeutical purpose. So analytically, exosome, it can be used in biomarker discovery, functional study, and pathogenesis investigation. And therapeutically, exosomes are valuable due to their high biocompatibility, strong barrier permeability, and ability to deliver content to specific site. Therefore, exosome can be modified and used to carry a drug, vaccine, and a regenerative medicine. So before we dive in this session, again, I have a question for the audience. So I'm wondering, what are the main goal of your exosome project right now? If you are working with exosome right now, is it biomarker discovery? Is it therapy? Or is this anything else? Again, feel free to type that in the chat box and please surprise me. Okay, so I will start with looking into biomarker discovery, and then let's see how this end goal will affect the isolation method of your choice. So a brief look at the biomarker discovery. Exosomes, they are ideal for this purpose because they can be found in easily accessible, minimal invasive samples such as blood and urine. Blood, urine, and saliva. And their lipid bilayer ensures stability and the preservation of disease-specific information. It also reflects the source cell. The biomarker, it can consist the cargo, like we mentioned before, the DNA, RNA, protein, lipid, and the metabolite. And there are some other characteristics like size distribution, concentration, and morphology. And all of those, they providing scientists with insight. So among everything I was talking about, RNA is actually one of the most studied area as indicated in this publication. So for today, let's pick RNA as an example to better illustrate what a biomarker discovery workflow looks like. So take this publication as an example. There's a starting material, either it's plasma, serum, or CSF. They are used. And then exosomes were isolated by a different method. The RNA biomarker was extracted and analyzed. Then the detection method showed either a up or down regulation in biomarker. And finally, after data analysis, this is used to build a correlation with a disease. So you see, the exosome isolation as boxed here is what we really care of. care about. But to choose the most suitable method, we really need to have a bird eye view of this entire process so we know which parameter needs to be prioritized for each specific objective. Well, for example, to do with a starting sample like plasma, purity is of top concern. Myself has mentioned that the soluble protein, it can co-isolate with EV. And because they are smaller and denser, well, good news, they are easily isolated using size-based or density-based method. However, lipoprotein, they are the non-vesicular extracellular particle. They can overlap with the blood EVs in terms of size, density, and molecular composition, which that will make purity harder to ensure in sample like plasma. So how do we go about it? Let's put a question mark here for now. And not only do we need to ensure purity, but when we look at a downstream application like the RNA detection method, you can see the method like microarray or next generation sequencing, they both mention that they need a large amount of sample to start with. Well, as many of you know, when you are dealing with the exosome study, the clinical sample like plasma, usually they just don't have a big amount of RNA to begin with, and therefore yield is another big consideration. So to optimize both purity and yield, take care of all the consideration, combining multiple isolation methods may be the way going forward. For example, this publication has suggested that utilizing multiple methods together, like combining ultracentrifugation alongside SEC or density gradient centrifugation. But what if I tell you there is actually a shortcut? Well, if you're a researcher watching this and focus on exosomal RNA, Norgen actually offers a complete workflow to extract your exosomal RNA directly from your sample in as little as 35 minutes. We support input volume from 50 microliter to 10 milliliter, and our proprietary technology is designed to specifically capture even the small size RNA at an ultra low concentration, and that is fully compatible with sequencing. And for something like exosomal RNA sequencing, you might know that we're dealing with ultra low concentration of a biomarker. And we need to make sure that we isolate all of them. And in fact, Norgen's method, we provide 1.5 times more mapped RNA read and up to 10 times more mapped microRNA read, which will significantly help you save time, save effort, and money down the road when it comes to sequencing. So if this doesn't sound amazing enough, I want to say that not only do we sell product to you, we also provide the isolation service alongside with our sequencing and bioinformatics service. So Norgen, we have been active in this field, especially when it comes to exosomal RNA for ultra low concentration, ultra low input sample. And for a limited time until the end of August, we offer 50% off on RNA isolation service, along with a free 6-sample pilot trial available upon request. If you are interested to learn more, feel free to scan the QR code over here, and I will switch the next slide in 5, 4, 3, 2, and 1. Let's go. So here is a big overview on the entire workflow that Norgen takes care of from A to Z. And not only do we provide a product and a service to help you achieve goal, if you have a specific R&D need, our contract R&D also have you covered as well. But enough about Norgen. Let's look at another application. So before I dive into the next topic, I'm checking the chat right now, and I see that, well, cancer research is pretty popular today. I see a couple of them in prostate cancer. I see biomarker discovery and a drug delivery. That's pretty cool. Well, today we are only focused on the analytical purpose, but given the interest in all across the board, we will make more content on the therapeutical purpose as well. So feel free. So please follow Norgen along. And before I dive into the next topic, so the next one we want to dive into is the functional study. And let's see how a different method or different research objective will influence your isolation choice. So the functional study, it's aimed to investigate how exosome mediate intercellular communication. And a common approach is to treat one cell type with exosome derived from another and observe the change in phenotype or gene expression. And to put things in perspective, here I have another publication as an example. And this publication, it starts with 3 cell line. There is a one metastatic breast cancer cell line, there's a normal breast epithelial cell line, and there's a monocyte cell line. So one of the experiments they did was to treat monocyte cell with exosome from metastatic breast cancer cell, and then later put exosome from the monocyte cell into the normal breast epithelial cell to evaluate the impact. Another experiment involved treating monocyte cell with exosome from both metastatic breast cancer cell and the normal epithelial cell to see if there is any phenotypical differentiation. So let's take a pause over here. Let's see what is one thing about the isolation method that is crucial here. Yes, the purity, the speed, the yield, they might all be important. But the most important metric here is that the isolation method, it must preserve the bioactivity and the morphological integrity of the exosome, so that those exosomes, it can achieve the functional as intended. And not all isolation methods can do that. There are isolation methods that inevitably destroy bioactivity and the morphology. And to achieve the goal that preserve both, we need an isolation method that avoid external damage, such as Norgen's method. In fact, this publication we're showing you here is actually using Norgen's isolation method to conduct their functional study. And let's get back to it and see how it performed. So in the first experiment, where the monocyte took up metastatic exosome and then released exosome into the normal epithelial cell line, you can see on the left panel that the exosome uptake was successful, observed under the fluorescent microscopy. And when you look at the right panel, you can see how monocyte experienced phenotypical differentiation under the influence of metastatic cancer cell and epithelial cell. And this effect was clearly observed too. So just like Norgen's method, size exclusion chromatography and polymer precipitation, they are both on the gentle end. Therefore, they are more useful when we are considering maintaining the bioactivity of exosome. And if you're a researcher care about bioactivity, you probably want to avoid ultracentrifugation and ultrafiltration. Although those methods come with a range of benefits, they do apply external force and may cause benefit to the exosome. Well, I'm not going through every single application today, but these are the 2 examples that give you the idea of how the end goal guide your isolation choice. And finally, here we come to our last point, the future outlook. Today, we touch on a lot of information for the first 3 parts of the presentation, and thank you so much for staying with me for this long. Now, let's give your brain a little rest with a story session on the history of exosome. So exosome, it was discovered more than 40 years ago, but we only coined the term exosome in the late 1980s. After around 20 years of study in the early 2000s, they began to explore as a vaccine carrier. As a Nobel Prize was awarded for exosome research in 2013, And the most recent event is that during COVID, exosomes, they were heavily researched for therapeutical purpose. And when we look at the number of exosome-related research papers on PubMed over time, we can see exponential growth. However, just to put things in perspective, when we compare the research volume to topics like DNA, exosome, they're still a very niche field. And what does that tell us? Well, this really tells us that the exosome, it represents a very new area of life science with strong momentum and it has the potential for major contribution. And that is why we want to look at the future outlook, because it's especially important now when you're choosing an isolation method now, and only by anticipating what the future holds, that can make you make a better decision in the present. And with that being said, here I want to show you two potential trends in the industry where it is heading towards. And the first one I want to start with is the combination method. And we previously discussed how purity and yield, they're competing goals, because purity means isolating selectively and yield means capture broadly. And to balance both, the scientists, they have begun to combine multiple methods. As you can see in the recent publication, the researchers combine ultracentrifugation with precipitation with size-exclusive chromatography, or even three methods together. However, only a few studies support this so far, and more work is needed to be done to standardize and optimize this approach. And another method that's gaining traction now is the microfluidics. Microfluidics operate on a small chip and allow only a tiny amount of fluid passing through it. This technique combines conventional isolation principle into a lab-on-the-chip format, as shown in the picture here, and the separation occurs on this micro scale. So although this chip-based format, it looks very different from the traditional method, the mechanism, they are actually quite similar, because those are all based on the physiochemical characteristic of exosome, and here is just a list of examples I want to show you. So those methods, because they use a very small sample volume, it is especially useful for sample that's hard to obtain, such as CSF. And it is also offer greatly efficient processing, precision control, and high sensitivity. However, microfluidics, it still has a very long way to go, and at this stage, the most publication, they are only a proof of concept, and the lack of standard protocol across different sample types that really limit commercialization. The biggest issue I want to point out today is that since the microfluidics, it really rely on the same physical chemical feature of exosome, the problem seen in the traditional method is still persist. And for example, the membrane clogging due to particle aggregation, it is a common issue in filter-based methods like ultrafiltration, and it remains an issue even at the micro scale. And I mentioned this not to discredit the potential of microfluidics, but just to provide a realistic perspective on this cool-looking technology. So let's take a break here and let's think, what does this combination method, a combination protocol, and the microfluidics, they tell us? Well, they really shows that the future of exosome isolation, it is multi-principle. It takes a combination of effort to move this field forward, and collaboration, it is at the center of this momentum, and that is a core value here at Norgen. For example, at Norgen, we have a specific isolation kit to complement Izon's size exclusion chromatography column. This kit, they come with Norgen's gold standard RNA isolation technology, which provide a complete RNA profile without inhibitor or any bias toward high GC content. This kit also specifically tailored for the researcher work with EV isolate using Ison's QEV column. So we can provide you with a smooth workflow. And that is just one example out of many. Over the past 25 years, Norgen has collaborated with numerous biotech companies to co-develop a product or help commercialize great concept. And all this is supported by our ISO-certified GMP-compliant facility. So, if you're interested in OEM opportunity or any type of contract manufacturing, feel free to contact us or scan the QR code for more information. And I will leave the slide here for 5, 4, 3, 2, and 1. And that comes to the end of today's presentation. So Norgen, we have been deeply involved in the exosome field, and we have a proven track record of supporting researchers through every step of the workflow. If you want to learn more about Norgen, we have infograph, a product roadmap, and a blog content available. And don't forget, we currently have this limited time promotion for 50% off RNA isolation service and a free 6-sample pilot trial run upon request. And that's the end of the presentation session. Thank you so much for staying with me, and we will now have a 2-minute break before we head towards the Q&A led by our PhD scientist, Dr. Alex Chauhan. Feel free to refill your coffee, do some stretching, and leave your question here in the chat box, and I will see you in a bit. Hi everyone, welcome back. I hope you have a great time. Just get your coffee refilled. And we're here with our PhD level scientist, Dr. Alex Chauhan.
Hello everyone. I hope you guys had a great webinar with Maggie, and I'm here to take more questions. I think Maggie can find some questions for us, I guess.
So we will go through the question as it coming, but feel free to still type in the chat box, and we will start with the first one. So Breathe and Drink 2030, welcome. And you asked a question, what kind of foreign bodies can be found in exosome? Pathogenic or heavy metal, for example? What do you think, Dr. Chauhan?
That's a very good question. Exosomes in itself can have a lot of things encapsulated in it. So imagine exosome coming out of a cell and it is kind of taking all the materials that may be present inside of a cell, which could be an outcome of an exocytosis. So it can have pathogens, it can have RNA, DNA, proteins, heavy metals, maybe, maybe not. It depends on how much uh, uh, relation it has with the exosomes. But I think pathogens we have observed COVID-19 with our during our pandemic that we had recently. We could detect COVID in after extracting exosomes and RNA isolation. We were able to detect COVID in that, and there are other people who have reported viruses and other pathogens, part of the DNA which is encapsulated, or part of the RNA that is encapsulated in the exosome. So yes, pathogens can definitely be found in exosomes.
Okay, thank you, Dr. Chauhan. I hope that answered your question. And now we're moving on to the next from Marisa. So Marisa has asked, does exosome from different sources like saliva or blood, does it show big difference in cargo profile? What do you think?
Yes, yes, this is a really good question. In fact, we had a study done recently in our lab. What we did, we were able to extract exosomes from plasma, serum, saliva, and urine from the same donor at the same time. And we were able to extract exosomes from them. Uh, it was very surprising to see that saliva had a very high difference in the profile as compared to that of the plasma and urine. Plasma and urine were kind of similar. However, saliva was completely different from them. So yes, if you are targeting a specific disease that you want to explore, and if you want to look at the expression of proteins or RNA related to a disease in saliva, which is maybe oral cancer or throat cancer or any kind of cancers that you're looking at, and if you're taking saliva as a sample, saliva is a better choice to take over blood or urine.
Okay, thank you for your answer and thank you for citing the story. That's really interesting for me. Hope that answered your question, Melissa. And now we're moving on to the next one from Robert. Robert has asked, what are the biggest hurdle in translating exosome research into diagnostic or therapeutic?
This is another interesting question. Yes, exosome, as Maggie mentioned very clearly, it's a growing field. It's a very new field, and there are several methods going that are being used to extract exosomes from different sample types, and everyone wants to take it towards therapeutics, because the biggest advantage of being using exosome for therapeutics especially is that it is more known to the human system. So whenever, let's say for example, you encapsulate a drug with exosomes, now the capacity of that going into the distant organs is more, because the body human system takes it in a better way as compared to that of any drug, which can be very toxic. So uh, the hurdles would be to produce the exosomes on a mass scale. The hurdles would be to obtain exosomes which are very free from any kind of contamination, free from any kind of predisposing factors that might cause other effects on the human body. So there are a lot of hurdles, yes, that would be affecting this, and it would require extensive clinical trials to get through these hurdles. Yes.
Mhm. Thank you, Dr. Chauhan, for the background information, and I hope that answered your question, Robert. The next question I got is from Zara. Zara asked, do you recommend combining silica carbide, which is Norgen's method, with other methods like SEC or immunoaffinity for better result?
I wouldn't recommend that, because Norgen's technology is very unique in its way. It does not have any bias, it just captures everything. Although it is having, it might not have as huge yields as you look into ultracentrifugation, maybe even SEC, but the quality, as Maggie mentioned in the publication, that the quality of Norgen's exosomes is amazing. The problem with this, it would be that when you, let's say for example, if you're using size exclusion chromatography and then followed by Norgen's SIC. The problem is the ones that you might have, the exosome types that you might have missed with size exclusion chromatography, you might not be able to capture with silicon carbide. The other problem would be that Norgen's kits are optimized to capture directly from the samples, not from the eluted exosomes. So if you're using eluted exosomes, let's say you had size exclusion chromatography and then you have eluted those exosomes in a certain volume, if you try to capture it using silicon carbide, the chemistry wouldn't work because the chemistry is optimized for a specific sample type. uh, on top of that, adding, for an example, immunoaffinity, it's even more specific. It will only capture those exosomes that has certain antibodies response, let's say for example CD63 or CD83. So if they are not able to, if certain exosomes are not having those surface markers, they won't be captured by immunoaffinity. So if your application is to capture certain uh, specific exosomes, then yes, go for immunoaffinity right away. You don't even have to consider Norgen. But if you want to look at the holistic picture, then I would recommend just to stick to silicon carbide.
Mm-hmm. Thank you, Alex Chauhan, for such a case by case explanation. So hope that answered your question, Zara. And now we're moving to the next one from Mike. Can Norgen's kit be used with animal model sample like mouse serum? What do you think?
Yes, of course. So Norgen's kits work with plasma serum. So let's talk about plasma serum exosome extraction kits. They can work with mouse serum. They can work with mouse plasma, human plasma. In fact, it works with any plasma or serum as far as it is cell-free. So the most important point here is to make those samples cell-free. If you have cellular contamination, then you might have other issues down the lane. So as far as it's cell-free, and if it's plasma or serum, it works with Nordic kits.
Mm-hmm. Thank you for the explanation. I hope that you answer your question. And then now there's a question from Liam. What are the challenges to isolate exosome from urine? So Liam is a urine researcher, I guess. What do you think?
Yeah, that's a very good question. Urine, especially in itself, is a uh, very widely used sample type, and everyone likes to work with urine because it is non-invasive. It does not require procedures like blood collection, so everyone prefers to use urine. However, urine sample collection comes with a challenge that you want to preserve the urine right away. There are certain ways you can preserve blood, but there are not many kits on the market that can preserve urine. Norgen has its unique urine preservative that can preserve urine at room temperature for several amount of days or even for a longer period of time. But that way you can also preserve your exosomes and you don't have to process it right away. The challenge that comes with urine exosomes is that you have to make it cell free as soon as possible. If you don't process the sample within few hours, then your sample might get degraded. Also, the exfoliated cells, if you don't take care of it right away, the exfoliated cells can burst open, which can release the cellular exosomes, which can skew your results, giving exosomal expression from the cellular content and not from the cell-free content, which can be a problem for your analysis. So these are some of the obvious issues that can arise for using urine as a sample type.
Okay, thank you, Dr. Chauhan. I'm really glad to hear that a Norgen's kit can actually help. Hope that answers your question. And we have another one from Hurara. Hurara has asked, if I'm doing proteomics rather than RNA sequencing, do I need a different workflow? That's really interesting. What do you think, Dr.
Well,
Chauhan?
not I. That's a great question, Maggie. I think these are really good questions that everyone is asking. We don't have any requirement or special requirements with our kits. If you're using, if your downstream application is for proteomics or RNA or other applications specific to exosomes, as far as you are able to extract exosomes using our kits, then you can apply it to proteomics or RNA-seq or PCR or using exosomes by itself. It works. So I don't think you have to need any specific workflow other than doing protein extraction from the extracted exosomes.
Sounds good. Hope that answers your question, Hurara. And Breeze and Drink 2030 come back. So the question is, I keep getting exposome and exosome mixed up. Maybe one is just airport, another is plant. It's really fun analogy. What do you think, Alex Chauhan?
Yeah, I think the key is to make it cell free. It would be great to further discuss these issues over a particular platform. Like, let's say we can get onto a call and further discuss this. We can really explain how to get away or how to work with your sample in a way that you don't get these kind of issues, like mixing up of exposomes with the exosomes. Basically, it's sample processing. If you process your sample in the right way, you won't have any issues of mixing exposomes with the exosomes.
Okay, thank you, Dr. Chauhan. Actually, we got another question from the same user. Thank you for being so active, asking questions. So also due to the time constraint, this is probably the last question we're taking today. And you're a lucky audience. So the last question of today is, how does air pollution affect exosome? And can it be, for example, transfer new nitrocyted DNA cell to cell?
Very interesting question. This is the first time I'm listening to something like this. Well, I'm not an expert of how pollution affects different aspects of it, but it is intriguing, and I would rather try to understand why can it even happen. So, in fact, it would be something for me to understand. Thank you.
Definitely, this definitely encourage further communication. If you want to ask Norgen scientists more questions, feel free to leave us a comment on the survey or contact us at info@norgenbiotech.com. We are very excited to hear from you. And I do see a lot of people are asking how many edits did everyone count. Well, I'm not gonna review the answer, but feel free to type your guess in the survey today. It is on the QR code right here. So with that being said, that marks the end of today's webinar. I'm really happy to speak with you all virtually, and have a nice day.